Hey, boys and girls, man, pigs, and heretics. A conversation between Immutable and Nestle. And we got some ground rules, though. If you guys talk about papers, please link them so I can put them in the description.
I guess so.
Because we don't want to pull a creationist, you know.
So, there we go, Michael. Great way to start a conversation. Thanks.
Yep. Sure. Well, fuck you very much, Immutable. Okay, you guys are on your own. Have a talk.
It's your role, not mine.
All right. So where do we start? Metabolism first, or? Well. Oh, hey, Leo's there. Hey, hello, Leo. All right.
Yep, Leo's here.
I think we can maybe start with you had a talk with David P. Neff on abiogenesis. Yeah, that's a very. Even though I have some disagreement, I still think it was a very good talk that you had. So
thanks, I appreciate it. Yeah. I try to consider the possibilities that I know of and try not to be too scathing of ones I'm less sympathetic towards or don't entirely understand fully.
Right. So I made on that stream I made some comments and
sure.
Yeah. Let's see. I at one point you start. Let's see here. Maybe I can show it. Or should I not show it, or should I show it?
You can share it if you want to. We can go through the objections or
sure
something like that.
It's a good start to maybe. So otherwise we don't know where to start.
Yeah, that's fair enough.
Yeah. See here, picking the right screen. There we go. Allow speed, please. Thank you. All right. So the first is so you at 8:50. You said that the RNA hypothesis is basically the same as the RNA first, right? Or maybe I'm.
Maybe I said that. I probably have said something like that.
Yeah, I think I clarified here that the RNA world isn't the same as the RNA first hypothesis. Like there's basically RNA world comes in two flavors: the strong RNA world and the weak RNA world. And basically the metabolism first proponents don't disagree with the weak interpretation of RNA world, where before DNA you had RNA. So metabolism first don't disagree with that, but they only disagree with the strong interpretation that RNA was the first biochemical, basically, yeah. Or maybe you don't have disagreement with that regard. So
yeah, I'm a little sympathetic towards RNA world in that regard, and that I could see it as conceivable as RNA being one of the first biomolecules that were present, and definitely.
So you mean the strong RNA? Maybe we get definitions confused. I agree with RNA world. I just don't agree with RNA first.
So RNA first, if I recall right, is essentially the it's kind of saying that RNA is the in some sense one of the first progenitor molecules, and kind of it runs the show in terms of all other chemistry that occurs after it.
Right. Yeah. It's saying I put it basically the Earth produces the nucleotides, and prebiotic chemistry produce like this genetic molecule, and that starts to self-replicate, and then later biochemistry is basically the invention of this genetic system made from RNA.
Oh, I think that's where we're going to disagree.
Oh,
because to me, I think my perspective is I think genetics is something that comes, if not instantaneously, it's one of the first things like that has to be established in biology, I think, or for kind of like this form of prebiotic chemistry that we're talking about. So I think that might be where we disagree.
All right. Of course, I'm a metabolism first proponent, so
yeah.
That was already a given, yeah. So
yeah. So my thing is, my argument is kind of like this, right? Metabolism is useful because it provides energy and it provides other building blocks for cells to use, and that's perfectly good. You don't need to worry about things like cell division or like replication for protocells, because hot and cold cycles near like a hydrothermal vent or some kind of similar situation is able to account for that, right? If it gets hot enough, you can have some type of cell division occurring. That's okay. My big thing, though, and I think it's a hallmark characteristic of life, is it's not that life just keeps on going. All life has in a way, and I'm going to regret using this term, it has a way of remembering, or it has a way of kind of drawing back on what was done previously, right? If a cell encodes a protein, right, and once that protein has reached the end of its ropes, the cell is able to create more of that type of thing. And to me, it seems natural that when you have a system like a protocell that's in whatever condition you're idealizing, that there needs to be some kind of mechanism that's preserving what information the cell is processing, right? And to me, I think that implies some kind of primitive genetics.
Right. It's basically a memory system in genetics. Like genetics remembers in the of course, and now of course it doesn't have memory in the sense that we have memory. I understand that.
Right. It's an analogous kind of.
Yeah. But although I would basically push back by pointing out that you view life as on the basis of the individual, but also you also, like in the talk, you actually point to a paper where it defines life in a systematic way.
Yeah. I think that'll actually. I think that's good to talk about a little later on, because it helps us kind of take a further step back and examine like what life is in terms of it being a physiochemical process. I mean, I think for now, what we probably both agree on is that no matter what model you want to take serious, no matter what model you want to endorse, right, there's definitely a notion of information involved in modern biology, and there's a way for that information to be expressed and drawn upon for whenever the cell needs it. That's done currently by genetics, and I think metabolism is something that comes later on. I think it's a process that emerges out of the activity of protein or of small peptides interacting with their environment, and kind of this essentially random dance of due to like phospholipid bilayers or lipid bilayers themselves being permeable to certain compounds, right? Different protocells will have different peptides, and some of those peptides may be very good at performing some basic chemical reaction or chemical transformation on that substrate. And as a result, it gives the cell a little bit more energy, and that process in turn results in, the cell being able to do more things. Maybe that means it explores more sequence space within peptides that it's able to generate. But that also may mean that when it divides, right, it's able to divide more often than other cells that are nearby it that weren't able to utilize that resource.
Right. But I still want to basically make the point about the definition of life on the individual level.
Sure.
Yeah. Like when you talk about like, oh, is this individual or at least is this object alive? You get a lot of problems. Like, for example, with is a mule alive or is a virus alive? Then you get like really into issues that get never resolved. It's basically an arbitrary distinction. But when you view life as processes, and also then it becomes I'm sorry, it's difficult for me to explain it in more detail, of course, for memory. I recommend the book by Eric Smith and Morowitz, The Origins and Nature of Life. And they make the argument that if you look at life from the ecosystem level, then that carries more of a consistent the life or the ecosystem carries the property of life more consistently than the individuals do. And what is consistent about the ecosystem level is basically metabolism.
Right.
Metabolism is conserved across the ecosystem level. In the individuals is a question about how metabolism gets partitioned with the control system of genetics. So to me that basically points to
Right.
To me that points to how
So I think what you're saying is something like this, that before we talk about protocells and things like that, there's this talk of say like atmospheric and geological processes that are occurring on Earth, that in turn, right, they have their own chemistry that's occurring, and in turn, right, that's kind of mimicking lifelike structures in its own way, because those structures are influencing their surroundings. They're generating certain chemical species more than other chemical species, and that's causing some kind of feedback cycle where eventually where eventually our notion of life, this notion of like biological life, develops out of that kind of system, which has been interacting long before essentially saying that, I think what you're saying is that these geological and atmospheric processes are contributing to the development of life before biological life is developing. Is that your point, or?
Like I'm not really sure. Like I'm just saying that what is consistent about the process of life. Like life is not really definable by the individual. Mm-hmm. It's more of a process, not a thing.
Oh, sure. Yeah. I definitely agree. I mean, I think. And
when you view it at the process or at the ecosystem level, then the consistent thing becomes the metabolism that is conserved. And then when you talk about individuals, for example, like us, like we have outsourced our own metabolism. Basically, like we don't fix our own carbon, we don't fix our own nitrogen. We need to eat other organisms for that.
Right.
And that result is basically how genetics partitioned the metabolism into different individuals. And so individuals is about genetics, and that came later when the ecosystem is being partitioned into different species that support the ecosystem. But before that, you have metabolism that organizes the whole system. So that's basically one line of logic that is in favor of the metabolism first idea.
That's really interesting. I guess for me, the different forms of metabolism that we see, this type of a distinction between chemotrophs and lithotrophs and autotrophs and stuff like that, I would say that that the way I'm viewing it being like some kind of compromise or some kind of a hybrid between RNA and peptide, first system would be that the reason we see different forms of metabolism is as these cells divide and as these cells develop things like proteins that allow them to move, like flagella, they settle into new niches that in turn bring about new forms of metabolism.
Right. Yeah, I think I have actually. I think I remember a good paper on like the early evolution of metabolism by the same author that I recommended earlier, Eric Smith, and also another researcher called Braakman, I think his name. And I will pull up the paper, and it basically shows how like the early divisions of life is defined by how metabolism changed in response to new niches.
Yeah, I would agree.
So I will bring up the paper. Like, give me a minute.
You're bringing up a paper.
Yeah, and it gives a very neat picture of this, and it also shows that one of the earliest metabolic networks or modes of carbon fixation is basically the acetyl-CoA pathway and also the reverse citric acid cycle.
Yeah, that makes sense. That's mainly how it gets generated nowadays.
No. Like nowadays, it's actually the reverse. Like we use the reverse, like the citric acid cycle, but the organisms back then used the reverse citric acid cycle.
Right.
Yeah, but actually, it should be backwards because we use the reverse and they use the original. So yeah, it's how. I will bring up the paper. Let me see here. Or maybe I can also.
By the way, Speed, if Dessel wants to join and he's able to, let him.
Yeah. Hold on. Did I bring
can probably add
right
some
paper?
stuff to this conversation that's beyond even
maybe
me and Nessie can contribute.
Maybe I brought up the wrong paper, but it has the same image that I want to show. It has the same image.
I could probably find it. Let me see. What's it called? The Emergence, early evolution.
Basically, what they show is that like the core metabolism that surrounds this citric acid cycle is composed of modules, and then later organisms they made small modifications to these modules, which gave rise to different forms of carbon fixation in response. For example, you have this branch here in cyanobacteria, which gave rise to like the Kelvin cycle.
Right. Oh, okay. I actually found the PDF. Here we go.
Right.
Let me see if I can get it to.
And if you go back to the Luca, you have like the network is basically composed of all these modules, but it's the reverse citric acid cycle and also the Woodlong or the acetyl-CoA pathway. Although they give a different name here, it's the THF4H4MPT sequence. But yeah,
here we go. So I can show it in better detail, I think. Let me see if I can. You're talking about this, right? This little diagram here, we're showing.
Okay, you can show, but I don't see it. Yeah, yes, that one.
Right, so it's talking about. Let's see. So they all seem to agree upon there being a common. Right, so the gray line represents the maximum parsimony trait linking the major metabolic phenotypes.
Is that from the same paper that Nestle showed?
Yes.
Yeah, it is. So let me see what's happening here.
I found it and linked it, so don't worry about it.
So what essentially it's saying is that it's explaining how, for most of these processes, the pentose phosphate pathway and
that's the Calvin cycle.
Yeah.
Or at least the pentose phosphate pathway is like the precursor to the Calvin cycle.
Yes,
basically,
that's right.
The Calvin cycle is the pentose phosphate pathway repurposed as a carbon fixation pathway. Yeah.
Right. So it's describing how there's a potential linkage between THF H4-MPT sequence and TCA. Huh. That is interesting.
Yeah, it's really complicated. Yeah.
Yeah, it does look like it. Do you want me to bring up the paper about Lyfe?
Oh, sure. Yeah. Go ahead.
Let me find it. Actually, wait. I can get a better version of it.
Also, I would like not potential join because
no, that's gonna.
Yeah.
We're gonna go down all sorts of different rabbit holes.
Be hours long. Yeah.
Surely you are kidding. You really think I would let him join? No.
We don't know, Speed. Sometimes you just go insane.
Yeah, sometimes I just get crazy. Anyway, link the paper that you're about to bring up, please.
Sure.
Or at least give me the title, and I'll hunt it out.
Sure. Let me title the paper in the description.
I also like the question he's saying, like the next big problem is how do you go from anaerobic carbon fixation to cycles which work aerobically? That's exactly the topic of the paper that I showed. Exactly.
Yeah, I think part of it though too is to
anyway, ignoring Otengio from now on. Ignoring him from now on.
Right. So this paper.
Oh, this one.
Yeah.
Yeah. Okay, you zoom in a bit because it's now really far.
Was that Bartlett?
Yeah, Bartlett.
Okay, so that was Bartlett 2020. Probably have that downloaded already. But let me go find that and get a link to it.
I put the name in the private chat of the stream, so you guys should be able to. If you paste it into Google search, you should be able to find it. But yeah, this is talking about defining life.
Yeah.
And it's talking about different pillars that we can use to define it. So the first major thing, I'm not going to go through the whole thing, but it goes through kind of what me and you were talking about, these kind of three major hurdles for life to go through, which is the first is replication. So you need to have a system which, first of all, has genetic material or some kind of material that's going to be replicated, and you need a process by which that material is in turn replicated. So some people take autocatalysis and specifically propose that there's RNA, and propose that there are RNA strands that, if you have a specific length, these RNA strands have the property that if they come across another RNA, they will in turn catalyze the growth of that particular sequence. So it acts as a way for other RNA sequences to kind of replicate themselves.
Right.
There's other solutions to it as well. What Nestlig is pushing for, metabolism, which definitely is important. So metabolism is a process where cells take some precursor molecule, it reacts in certain ways, and in the process of doing so, it forms energy.
Yeah.
As the result. And compartmentalization. So obviously, cells have an inside and they have an outside, right? There's cell membranes that cells have that separate their internal chemistry from the external chemistry of the environment. And one thing that you have to account for is number one, how do these compartments form, especially if you're going to say especially in regards to especially in regards to say compartments that are present within organelles and stuff like that, because that's a process where it's a bit delicate since you usually need peptides or proteins to do that. And so these are three properties that life always
we're
has.
talking about organelles in eukaryotes, or are you talking about like organelles
even in bacteria, right?
Yeah.
So like hydrosomes and things like that.
Hydrosome? Is it an organelle in eukaryotes, hydrosome?
It is, I believe.
Oh,
It's a
maybe
way
I-
for cells to eliminate excess water, I think. I could be dead wrong, though.
Let's see here. Hydrosomes. Or maybe I'm confusing something else. I think I'm confusing
It might have a
something.
different name, but that's the name
Yeah.
I'm used to it having.
All right. But yeah, it's although yeah, I get your point about that. It's compartmentalization is of course also important.
Right.
Although, I think for example, you are familiar with the alkaline hydrothermal vent hypothesis as well. That also provides some of the compartmentalization that kept the system in place at the same time. Yeah, before the lipid compartments were there.
Right, and this paper goes on. So it talks about the general kind of thing that happens in Origin of Life. There's two main. Excuse me. There's two main camps that try to tackle this problem. One is bottom up, the other is top down. Bottom up, we try to take basic chemistry and physio and physical processes, and we see if we can make life given known chemical reactions and things of that nature. So we try to avoid kind of looking at the back of the book to see to kind of get influence from the chemistry that life does use currently. But top-down approach is essentially trying to infer the properties of life given what we have currently, and you can use that by doing phylogenetic analyses of certain genes and the proteins that are embedded, and trying to.
Yeah. Oh, so sorry. I was saying that what the paper that I showed previously is what they did the same thing instead of genes. They did the same with metabolic networks, and they showed that how different metabolic networks relate to each other and connect to a primordial metabolism, basically. So yeah.
Yeah, I think that's going to be true no matter how you start. No matter how life starts, there's probably going to be one form of metabolism that takes place, but it diversifies as cells encounter new materials and as the need for new niches begin to develop.
Yeah, and we all organisms like us, we reverse the citric acid cycle, going backwards than the original. Yeah. That's a good example. Yeah. Okay. If you want to show more, I have one particular section in the paper that I want to.
Sure.
Also,
do you want me to?
Scroll down a bit. Yeah. Scroll down a bit. Scroll further down. I think it's just after the list of points where they. Yeah, just after that. Let's see here. Actually, I don't remember exactly where it is now. Sorry. Can you search for the word system? Search for the word system.
Well, it appears all 96 times. So that's
damn it! I will. I have also the PDF, so I will look for it and then come back. Give me a minute.
Okay. Yeah, while that's happening, I'll go through the last thing I kind of wanted to touch with on this paper, which is the processes it's using to kind of to characterize life as a general characteristic of the universe. The first is dissipation. So obviously, what the second law of thermodynamics means and why life exists to begin with is that life is a system that drives Life is a system that supports entropy. It's a system that takes complex molecules and turns them, or rather, it's able to utilize complex compounds internally and to transform that into compounds that are entropically favorable, such as hydrogen and carbon dioxide and things of that nature. The other one is autocatalysis, this notion that if you have certain reactions under certain conditions, or rather certain compounds under certain conditions, they will begin to react in a cyclic type of manner, which we've observed that due to Lee Cronin. The fourth, homeostasis, so the ability of cells to, the ability of a system to regulate its internal environment from its external environment, sure. And the other is learning. So the ability of a system to kind of take information from its, to process and take information from its environment, and then in turn, kind of use that information in a way that's productive to the organism. So that might mean an organism mutates a protein that has a higher turnover rate for some reaction, and that particular phenotype or that particular change to the protein gets passed on into all the descendants of that particular organism.
Got a question here about metal hydroxides, zinc. Lots of active sites in molecular biology.
I found it. Can you
Sure.
go to the page 8 right at the top? Page 8, right at the top.
Right, page 8. Whereabouts?
Or maybe starting just before 8 and then the paragraph. Yeah. Oh, yeah. We take this moment. I think. Oh, the next the next paragraph. We take this moment.
Okay.
Yeah.
So it's saying we take this moment to emphasize that our definition of life applies at its simple, at the system level. The pillars can be ascribed to a certain system, depending specifically on the boundaries which we use to refer to that system. For example, which pillars do viruses perform? A single virus in isolation cannot perform many of the pillars. Viruses in a system containing viruses, bacteria, and nutrients can perform autocatalysis and through the coercion of the bacteria host. Yeah, okay.
Go on to the until the paragraph is finished.
Okay. So it's saying a single virus in isolation can perform many of the pillars. Viruses in a system containing of these viruses, bacteria, and nutrients can perform autocatalysis and through the coercion of their bacterial host dissipation. Viruses in a biosphere that is evolving with the environment will cause not only autocatalysis and dissipation, but learning through evolution as well. In certain ecosystems, viruses may even impart homeostatic attributes to the system by introducing auxiliary metabolic genes to their hosts and recycling organic matter via lysis. Okay. Thus, the argument about whether a virus or a mule, to cite another classic paradox, is alive becomes irrelevant under the assumption that lifeness does not emerge at the molecular or cellular or organismal level. Like others have suggested. Like others have suggested, we contend that the living state may be assessed at an ecosystem or planetary scale.
And you can see like others have suggested when they cite 46 and 47. One of those is the book that I recommend.
Okay, interesting. I'll be sure to check these out.
Yeah, The Origin and Nature of Life by Eric Smith and Horowitz. Yeah, they make the argument that I just made about like at the ecosystem level, life seems to be more easily definable than at the individual level, and individuals is more of a consequence of how genetics later partitioned the metabolism, which happened before. But yeah, that's basically one of the logic. But I also have some commentary. Yeah, I have also commentary about your previous comment about the dissipative nature of life, like the law of the second law of thermodynamics. You know, right?
Mm-hmm.
It's also something that the metabolism first goes into. Like one of the criticisms by them says that the scenarios for like the RNA. I heard some feedback. Sorry, from somebody.
Yeah, I have no clue.
Thank you, Speed. So basically, they point out that at the RNA scenarios, like for example the bond scenario where you have RNA replicating, that is basically thermodynamically flat. It doesn't. There is no drive. There's no disequilibrium that drives the process of life around. So if you go by like, oh, life is a dissipative process where it takes energy, like it takes energy from a disequilibrium from its environment and dissipates it, and that dissipation drives the whole system, which is why life is thermodynamically favorable. That doesn't really happen with the many scenarios proposed for genetics first, but it does happen with metabolism first. Like, let me ask you a question. Like, do you know what type of disequilibrium life relies on?
Usually
What type of
electrochemical.
Right. It's redox chemistry. Like you have one chemical that has electrons to donate and one chemical
One which
that has
accepts.
yeah, exactly. So of course, different life forms use many different redox pairs. Like for example, we, for example, use organic or carbohydrates as the donors, and the acceptor
Right.
is oxygen. But of course, that system came later because oxygen didn't exist at the beginning. So what life early used? Well, like I mentioned before, the acetyl CoA pathway, which involves the reaction between carbon dioxide and hydrogen. And the beauty of that is that without life, like there is energy to be gained when you react hydrogen with carbon dioxide to form water and methane. Although that's actually a reaction. Even though you would think, like, of course, methane reacted with oxygen, it also produces energy. But when you have hydrogen and carbon dioxide, then producing methane is favorable. Although there is a huge kinetic barrier that prevents this from happening. So what metabolism specifically basically does is it forms a dissipative channel that relaxes this equilibrium between hydrogen and carbon dioxide. So basically the formation of metabolism to form a channel that allows hydrogen to react with carbon dioxide is what gives rise to life. That's one of the
I think the really curious process about that too is, like, if you look at the so something I'm a little more familiar with is, say, like nitrogen fixation, which is just as hard to do as say hydrogen fixation. What, like, the process that occurs under, right, is a very complicated dance and very complicated chemical process. In terms of, having these very interesting organometallic systems like that involve like molybdenum and certain other things.
Right.
And the thing is too, right? This is a general characteristic of metabolic systems, and not not needing like to go to a particular example, is that the point, like, of a catalyst in terms of normal chemical reactions is that catalysts are not chemically altered over the course of a reaction. Right? If you start with a catalyst, you end with the same catalyst over the course of the reaction. Now, biology does obey that, but it plays a little fast and loose with that. What do I mean? Well, if you're using certain chemical reactions, what life will do at times, if there's things which are bound to a metal, say like histidines that are interacting with a metal or something like that, is the metal will often act as an electron acceptor, and at some point later it'll it'll use that gradient to kind of return electrons back to the system. But remember that there's this process of metabolism, whenever it develops within protocells, right, is if something happens, say to a cofactor or say to some part of a peptide or protein that's occurring, right, in order for a reaction to take place, there also has to be simultaneously a process that's allowing for that system to return back to its state at rest, right? So if you've taken a hydrogen away from an oxygen atom over the course of a reaction to make it occur, or something like that, there has to be a hydrogen that's given up somewhere in order to regenerate the catalyst that was used.
Right. Exactly. So yeah, it's also what metabolism first goes into as well. Like you made, like with the nitrogen fixation example that you gave, you mentioned that the protein that is responsible for that reaction has a metal center.
Yep.
Right. So yeah, that's also interesting when it comes to proteins in general. Like most proteins, or at least half of them, have what is called a cofactor. Like they contain, in addition to the protein itself, they contain something extra that is involved in the reaction as well. Like, and in the specific example of nitrogenase, they contain a metal center that is iron, sulfur, and also molybdenum. And interestingly, like when you go further into core metabolism, where you also, for example, the pathway that I previously mentioned about the acetyl-CoA pathway and also the citric acid cycle, these reactions involve proteins that have these metal centers as well. Many of them iron, sulfur, and also nickel, iron. And what the metabolism first proponents point out is that it seems that these reactions it's almost like the proteins later enveloped, or they enveloped the previous reactions, which is basically geochemistry enveloped in proteins. That's what it seems like to them.
Huh. Now that is interesting.
Yeah.
So in that case, the argument would be that proteins are one of the last things that comes along in terms of
cofactors preceded proteins, basically, is the argument. Especially the metal, especially the metal, which seems like they are minerals, basically, which are inside of our cells. So yeah, mineral chemistry.
There we go. I guess I would still ask the question, and maybe it has given, maybe there are answers that are that have been given to a satisfactory level. But in terms of I could give you that proteins are probably something, or might be a thing that comes along later in terms of charting out a path for life to get where it needs to.
And it's also interesting, if I may, or maybe do you want to finish first? It's also. Sorry. And it is also interesting about even proteins that don't have cofactors. They use special types of amino acids that is involved in the catalysis. And these amino acids are synthesized from the same pathways that make cofactors. So basically, they are cofactor in amino acid form.
Huh, that is interesting.
Yeah.
My question was it was in regards to and the answer probably is going to be yes to some extent. But the question is, so where's the union between metabolism and genetics?
Right.
Where is genetics coming into this equation?
So I mentioned, like, of course I cannot give the full detail because that is still a very complicated subject. Like the transition towards genetics is a huge gap to resolve as well. But if I can bring some points to it, like if we start with the core metabolism, like for example the reverse citric acid cycle, one of the branches that comes from that is the synthesis of the like the first nucleotide, like ATP and GTP, but there are also many other products that come from these, like for example, ATP together with a amino acids forms the coenzyme A, cofactor.
Right, acetylcoenzyme A.
Yeah, that is produced from cysteine and ATP. There are also many other cofactors that are produced from these nucleotides as well. So what it seems to me is that first you have these small metabolites from the core of the citric acid cycle. Then you have these cofactors, which supports metabolism before proteins. And then later, it's also interesting about the cofactors. They show the beginnings of polymerization. Like for example, NADH is a combination of two nucleotides, NADH. So it begins to show the first moments of polymerization. And then what it seems like then later you had, I don't know the specifics of course, but then you had more polymerization, at least more organized polymerization, where you only polymerize nucleotides together, and of course peptides as well separately. But that is where I draw a blank. I don't know the specifics when it comes to that. I think the book that I recommend goes in more detail about that as well. How RNA, so how RNA and nucleotides start to cooperate to form polymers, very complex polymers later on.
Yeah, that's very interesting. I'll look into that because to me that's always been a really like kind of
it's like the cofactor world also. Yeah, the cofactor world also seems to show that like RNA nucleotides and amino acids also formed a cooperation. Like many cofactors are a hybrid between amino acids and the nucleotides as well. So that it also shows some form of cooperation before they went into the polymer phase, basically. Yeah.
You know, this is a little bit of a weird tangent, but and maybe it was different back before, when things were very early in life. But one thing I think that's kind of universal with genetic systems is that if you're going to like whatever genetic content is going to be used, it definitely seems like adenine and thymine are going to be the two nucleotides that come along last, if anything else. And the reason for that is if you have a system that uses cytosine and guanine for base pairing, especially in a system like near a hydrothermal vent or hydrothermal field or something like that, well, it takes more energy. Essentially, there's more stabilization that can be conferred by using cytosine and guanine rich sequences instead of using adenine and thymine rich sequences. And the weird part to me is that why are these cofactors using cytosine so strongly and so preferentially instead of using other nucleobases?
I think like most factors also use ATP, I think, right? As the base.
Yeah.
Yeah. Like ATP itself is a cofactor too. Yeah.
Right.
Yeah.
To me, that's a little bit interesting because it seems like it would be strongly leaning towards cytosine or guanine in that. Well,
yeah. I think like there was a recent YouTube presentation by Nick Lane. I can look it up where he also mentions why ATP is preferred over other nucleotides. And let's see here. I can look it up. I don't remember exactly what the reason he gives, but you can watch the video.
Yeah, Nick Lane is actually somebody I do want to look into more. Because a lot of the stuff you're saying is reminiscent of the stuff he's
I first came across his work, and then later I came across the work of Eric Smith. Like he's also a very, very good person to look up for this stuff. Like Eric Smith is really he has a really elegant way of and like elegant vocabulary where he talks about anything basically, anything. You can listen for him for hours and you'll not get bored.
Oh, Tangelo asked a good question.
Oh, Oh, that's a very rare event.
Yeah.
Why? Well, because part of it is going to be, even if there's a million different nucleobases and nucleotides that can form, right? Some of them are going to be stable in some environments. Some of them are going to be not stable in other environments. And part of it as well is what you have to consider is when a system is able to undergo chemical synthesis, how easy is it for the system to synthesize cytosine in comparison to some other weird nitrogenous base that'll get glycosylated?
Yeah, cytosine is fairly simple as far as
Yeah, it has, what is it?
NH2 and a
compound.
double bonded oxygen. And
Yeah,
there's
an amine,
yeah.
an amine and a ketone.
Yep.
It's not using anything overly fancy.
There's also like about the there's also something else I can mention about the metabolism first in relation to the genetics. Like you know the genetic code, right?
Yep.
How the different codons correspond to different amino acids.
Right.
There's an interesting correlation with which types of codons correspond to which type of amino acids. Yeah. Okay. Do you know which? Do you know what correlation?
Yep. Mm-hmm.
What is it?
GC-rich codons. GC-rich codons code for some of the more important amino acids responsible in protein architecture. Those being
Right.
proline, alanine, glycine. I think glutamic acid and I think lysine are usually coded by them. I could be wrong though.
Yeah, there is actually a stronger correlation if you look at the biochemical pathways of these amino acids. Like the first nucleotide of the codon correlates to the precursor, the biochemical precursor of the amino acid.
Now that is interesting as well.
And the second one I think is correspond to a certain kind of chemical reaction. Like if of course I don't know the specifics, but if it is for example, if the second codon is a T, it's hydrophobic. If it becomes A, it's hydrophilic, and the others are neutral, for example. So it's like in a very tight correlation between how the amino acids are made with the genetic code.
Right. Yeah, that is very interesting too.
And I can actually can bring up a good image that shows this. Let me see here. Share screen.
Tangelo's just losing his goddamn mind in the chat, of course.
I don't care.
Nor do I. It's just fun to watch.
Yeah. We are having a productive conversation.
Confrontation. Wow, okay.
Yeah.
Fucking shit.
No. When I talk for too long, I start to ramble. It's just me. This is a good review paper on like the metabolism first perspective.
Yeah. If you want to pop that into the thing, I'll happily look at it.
Yeah, although it's behind the paywall, but you can of course use sign
up
as always.
I know how to find it.
See here.
I will link it for you in the description.
And if you go a bit further down, it talks about the evolution of metabolism pathways as well, and here is the acetyl-CoA pathway that I mentioned before. But about the genetic code, let's see here. Where is it? Ah, come on. Where is it? It's a very long paper. Yes. There you go. Maybe I can zoom up a bit. So what it shows is that these above here are G-coded. I'll zoom in a little more now. So there we go. I can show the full image then, but anyway. So these are the G-coded amino acids that are derived from alpha keto acids, and these are the C-coded amino acids derived from alpha ketoglutarate, and these are A, U. And what's also interesting is that the like the special types of amino acids that like for example histidine, and these are the ones that I mentioned about. These are derived from cofactor synthesis. And what basically this shows is that these amino acids came later. Like evidence shows that the amino acids, how do you call it, repertoire used to be a lot less than we have today. Like for example, like I think the early ones are like 16. There used to be only 16 amino acids, and then later, as life became more complex, then another few became added to it. So,
What
yeah,
page is this on, if I can ask? Sorry, what page is this on, if I could ask?
oh,
I think I see it. Page 18?
Yeah,
or something like that?
yeah, 18. Yeah, I think so. Yeah.
Okay, that is interesting. So what it's saying is, decoded amino acids derived from reductive animation or transamination of alpha keto acid. Ah, that is interesting. Huh, now that is very interesting too. Huh.
Like for example, like it shows here like histidine doesn't really have a correlation regarding the nucleotides, maybe late evolutionary addition, and the aromatic amino acids here also important in catalysis. So yeah, they are like a bit of compared to the other amino acids, it's a bit of outliers when it comes to this correlation. So it seems
that
to,
is,
yeah.
You know, the really interesting thing I noticed, and it points this out too, is that the aromatic amino acids appear to be really really late coming in terms of their appearance.
Yeah.
Now that is interesting. Huh. All right. Well, at the very least, something to think about. That's interesting.
Yeah. Of course, don't take my word for it. I'm just a random person on the internet. I just recommend good books and good papers.
Hey, I mean, you know, to borrow phrases, to borrow adisms from Speed. I'm a whore for good theories. So, you know, I will happily consider
a theory slut.
A theory slut. Well,
yeah.
I guess I have to put my own spin on it. So.
And there's also another thing interesting about this paper. It goes into like I didn't know this before, but when I read this paper, I was shocked at how different pyrimidine synthesis was compared to the other ones. Like how do you call it again?
Purines,
pyrimidine and purines. Yeah,
purines.
Okay, yeah. This is basically the how. I guess this like a here are the purines, and on the right are the pyrimidines, and they are very different chemical pathways.
That's that same paper? What page is that?
The same paper, yeah. It's page 25.
Wow.
I wonder if Gur will do any streams tonight, since Otangelo was just fuming at the fuming at the lips.
And notice that the precursors, many of the precursors for the purines and pyrimidines are amino acids. All of them, almost all of them.
Yep.
Yeah. There's one interesting here. You see this one? 4-metyl tetrafolate.
Yeah, 4-metyl tetrahydrofolate.
Yeah. That is involved in the acetyl-CoA pathway.
Now that's interesting.
Yeah. It's I didn't know.
I really do. I wish organic chemists would do this more often. Where if you go back up a little bit, where it's showing like purines and pyrimidines, and it gives like different colors to the atoms.
Which one?
To show how they're all connecting. The one you were on previously.
This one. This one.
Yeah. There we go.
Yeah.
It's nice how it lays that out, because I'm looking at it and I'm like, okay, what's reacting with what, and then it shows the.
Yeah. Of course, there are multiple steps involved. Like I think.
Definitely.
Yeah, but they look at basically, where does this nitrogen come from? It comes from this glutamate.
Yeah, where is this nitrogen taken from? Oh, it's taken from that atom. Oh, where is this oxygen taken from? Oh,
and it's also interesting, like the point where nitrogen enters the biosphere, basically the whole biosphere. It's just one particular point. It's the synthesis of glutamine and glutamate.
Right.
And every other compound that has nitrogen is made from that, or eventually by a long pathway from that amino acids. That is how amino acids becomes assimilated into the whole biosphere. It's one point in the metabolism, and that lies very close to the citric acid cycle.
Huh. That is interesting. What I would be really interesting to see happen is I would really be interesting to see so like the proteins that catalyze these reactions nowadays. I would like to see if there's if you could do something like, for instance, where phylogenetic like predictions and potential reconstructions of the most ancient versions of these proteins that would have existed, say, in Luca or or in one of the first life forms, and see how that protein behaves.
Funny that you mentioned that is that I actually read a few papers. I read a few papers that showed that, like, excluding the later amino acids, the special ones.
Yeah.
And also the earlier ones, they are able to produce these protein folds, which are predicted to be the oldest protein folds. I think the one of them is the Walker A motif, and that binds to nucleotides.
Huh. No, that's interesting.
Yeah, and also another paper shows that random polypeptides, not even proteins, random polypeptides are able to bind to metal cofactors, for example.
Yeah, that makes sense.
Yeah. So what it seems is that even the earliest proteins are basically able to bind and stabilize the cofactors, and that is the beginning of how the cofactors became enfolded into the protein world. Well, at least to me, what it seems to be like.
Yeah, I mean, I think the other thing too is kind of how easy is it to get the biogenesis of these cofactors?
Yeah.
How easy is it for these cells to create them? And obviously, in the case of like our like current form of life, eukaryotes don't necessarily need to produce all of them.
Right.
Certain forms of life would need to be able to.
Yeah, it's actually very interesting. Like about nitrogen fixation. No eukaryote is able to fix nitrogen natively. They are always reliant on some symbiotic organism. A prokaryote. Symbiote.
I think it's pretty clear from this diagram. Right?
Yeah, just look at it. How can you argue with such clairvoyance and clarity like that? I mean, there you go, Tangelo, with the whole freaking intelligent design thing. What's what's happening? Where to look?
Yeah. Yep. But there's also a very interesting thing about this here. Like even among all these different tendrils and lines, like if you follow any line to anywhere, they all end up at the citric acid cycle. So everything is made from these small molecules that compose the citric acid cycle. So
I gotta show that off. A pharmaceutical company sends those out for free, I think. I think it was it Roche? I can't remember, but I went onto their site and they sent me 2 gigantic posters. One is metabolic pathways, and the other one is cell molecular processes, and they're both like 4 feet across. And you still need a magnifying glass to look at the details. So pretty damn cool. So
yeah, definitely.
But they're so big, there's no place to hang them except on my whiteboards, and now I can't use my whiteboard, so I'm really in kind of a pickle here.
You poor thing.
I am a poor thing. Yep. Both my huge whiteboards are covered up, so screwed now. So what else, guys? What do you?
I don't know. I think we've kind of hit a interesting point where I think we've both presented really interesting points. Dessel would be an interesting person to have in the stream because Dessel, I don't know, maybe
He-
Dessel does hold to metabolism first.
He was heading out, so he couldn't join.
Of course
So-
he is. He has a life and all that. Yeah.
Yeah.
Okay.
If there is one final thing that I want to mention about this, is that like if metabolism first is right about life is basically the solution to make hydrogen react with carbon dioxide. Like without life, this equilibrium is never resolved. So basically, life is the solution to this problem, and that suggests that life is inevitable under the right circumstances. It isn't.
That book is Smith and Morowitz, right? Is that, or is it Smith and? Is it Morowitz? Was he the other author?
I think Morowitz, yes.
Morowitz, yeah. I've told you about this book a couple times. Immutable.
I think so. Yeah, definitely, Speed. You've been really hounding me on the abio topics to to look into and to hammer down on. That's definitely been you.
Actually, I haven't been. No, I've been working on.
I have to warn you. It is a pretty heavy book, both in terms of number of pages and also detail and technicality. Yeah,
it's not an easy read, not at all. It's like what 800 pages.
It's not a layman-friendly book, but for people like you and me, I think it's a really good book. Yeah.
Yeah.
Like if you know
that's
somebody,
the thing too.
Yeah.
Like there's that's the thing as well. Like a lot of people who want to argue about this, like it's hard for me too. I mean, especially because I'm doing courses and stuff. But there is a genuine there is a lot of like work that has been put into this field and a lot of stuff to understand to really to really kind of to try and resolve this question of how life as a phenomenon works and also how it formed, and more importantly, why it formed as a process.
Oh damn it! Let me
I'm also trying to find some papers that I mentioned about the oldest protein folds. Let's see here if I find one. Like there are several of them, but I think I can find a very good one about this. Let's see here.
There it is.
So, okay. Apparently Otangelo's arguing that base pairing has nothing to do with stability and heritability. Yeah, there we go.
It's amazing if you say anything about the origin of life, Otangelo somehow hears it no matter where he is.
I know, it's like fucking it's like whispering,
It's almost like like God told him.
It's like a dog whistle. I mean, golly, he just keeps coming back and same fucking time over.
Here you go. So here's the book. Let's see if I can get it maybe a little bit bigger, but anyway, it's kind of cool because they propose life as sort of a almost like it had to be, like it's an extension of geology, and that's one of the things that they get into in the beginning. It's pretty cool.
Yeah, that's
another argument. Right? Sorry, that's another argument for the metabolism. For like they show a continuity from geochemistry to biochemistry, basically. Yeah.
Yep. Oh, and I suppose I got to put this in the description too, don't I? The forced geosphere.
I'd be curious to see if it's arguing that all types of geochemistry converge upon this type of solution, or if it's only specific types of geochemistry that are familiar, like that the Earth uses or that the Earth did use, like way back, like during the Hadean period.
I think one of the geochemistry that is involved is serpentinization. I think you're familiar with that, right?
Yep.
Or not? Yeah, it's like how water reacts with a certain type of mineral, and that releases hydrogen and also many different kinds of reduced chemicals as well.
I've always been impressed by inorganic chemistry. It's When I was
It's
taking
incredible.
when I took inorganic chemistry, we were being taught it by an organic chemist because we didn't have an inorganic chemist present at the time to really teach that subject in as rich detail as it could be. But one of the most surprising things is that like a hydrogen or a methyl could be a leaving group in inorganic chemistry. Just absolutely amazing.
Yeah, this book is pretty intense. I've tried it a few times.
It's like I think the biggest issue is like you have to read it a few. I feel with through some of these books like you read it like 5 times before you understand it once.
Yep. Yeah, it's pretty cool. But it is packed full, 660 pages. And
if only it was 6 pages longer, then we could really piss some people off.
Oh, 666, yes. Have to check on that. Okay. So what else? So should we end?
I think that's good.
I will I'm almost done finding the papers, but of course I can give it to you after you end the stream. So yeah.
Okay. I'm gonna let's see, Nonenzymatic. I think I have that one. I already had that one linked in the description. The one you just put in private chat.
Like I mentioned more about the all those protein folds, and I will give it to you in the private chat after you end the stream, and you can add those in the description later. Is that good? Okay, good enough. Okay, you ready to shoot in the head? Sure.
Shoot in the head.
We're gonna shoot it in the head, guys. So thanks all of you out there. 301 chats. This was pretty active. So
yeah, this is amazingly active.
Yeah, let me capture those chats before YouTube screws me up here again. Let's see. Do I have the auto? Yeah, the auto thing off. Okay, got it. Okay, guys. Goodbye, cruel world.
All right. Goodbye.